this hypothesis was been discarded by the scientists that because the metal carbides are unavailable under the Earth's surface, so the inorganic theory was discarded, and this theory mainly consists of two main hypotheses, that is the Mendeleev theory and the cosmic theory. But nowadays, the organic theory is the most studied and the most considered theory. In this theory, the dead remains of the plants and animals under the marine surface get buried and decomposes into simpler petroleum hydrocarbons, and later of millions of years they are extracted as the petroleum crude oil. According to the poster, if you will see the topics, first is the chemical composition of the petroleum hydrocarbons. The petroleum hydrocarbons are mainly composed of alkanes, aromatic groups, and the organometallic compounds such as vanadium and nickel. The bioreactivity of the petroleum hydrocarbons, that's the petroleum hydrocarbons can be reacted fastly and very vigorously with the living organisms like bacteria, fungi, or some viruses also. The molecular characterization of the petroleum hydrocarbons states that the petroleum hydrocarbons can be degraded using living organisms, and the living organisms will chooses from the environment and the surrounding where the petroleum hydrocarbons are present. This thing is done mainly by the PCR cycle. Here the PCR cycle means that the polymerase chain reaction. In this, a single DNA which contains two strands gets separated at the temperature of 90 to 94 degrees Celsius and then converted into two single strands. And after that, if we cool down to the 50 to 68 degrees Celsius, and the process of annealing, in this, the DNA primer, that's the initiation of a DNA, gets attached to it, and then after attaching to two DNA primers to two different strands, then if we will heat up to 72 degrees Celsius for the elongation process, the nucleotides of the DNA get attached to it and they form two complete different DNA of the same species. In this process, we can make two DNAs from one DNA, and if we repeat the process, the DNA will be formed millions in numbers from a single DNA. This PCR helps in the degradation of hydrocarbons into simpler substances like CO2 and H2O, and this PCR cycle identifies that what type of microbes or microorganisms is going to react with the petroleum hydrocarbons and what factors are they going to be affected, which will be later discussed in the chat. Then if you are moving to the second topic, that's the aerobic and anaerobic processes. The aerobic degradation means what aerobic means in the presence of oxygen. The aerobic degradation consists of that the petroleum hydrocarbons get degraded by the microorganisms in the presence of oxygen, which convert them into simpler substances like CO2 and H2O, which is less harmful to the environment than the petroleum hydrocarbons. In the same process, the anaerobic degradations consist of different groups or compounds like sulfates, nitrates, and other compounds. In this process, the harmful substances are less, but in comparison to petroleum hydrocarbons, they affect less to the environment. If we see to the graph that on the release of the petroleum hydrocarbons, the air release is the maximum, which is affected to the living organisms, but the marine species which are present are affected more because the water is dissoluble in water. So to prevent this only, if you have studied about the bioinformatics, the PCR cycle is a tool of bioinformatics. So, as per I have studied on this topic, I have got that the PCR cycle is a tool of bioinformatics. PCR is completely a wet technique developed by Carrie Willis for amplification of DNA. Amplification means what? DNA तो आपको multiple copies बना होगा, जो microtubes. पर DNA vector protein जो TC9, TK42, इनकी तरफ होता bacteria जीनोम पड़े आसपास, तो infection हो जाता है. Method use करो, protocol use करो, तो टेलीफोन पड़े आ जाता है, फिर पुराना टेलीफोन, तो DNA पहुंचता है नहीं. तो bioinformatics एक तो लगेगा bioinformatics study of data, protein structure, data mining, disease prediction, genome sequencing, that लेकिन तारो entire topic को पूरा knowledge. Sir, genome sequencing को हमें use करेंगे different living microbes वगैरह को form करेंगे, degrade करेगा और petroleum hydrocarbon. कैसे करेंगे? अभी वाला कैसे करेंगे? पत्ता और नाला का बहुत ज़्यादा पानी, है ना? पूरी कहने जो रोड का जो तार और नाला का पानी, आओ तुम्हारा जो ये जो आगे तो देगा तो जो लाइट काम करते जो cross exam नाला पानी. You think microbes are different or you think microbes are same? So according to the environment, there varies from the place. But microbes are still equal. Adapt करके जो नहाले तमें yeast पकेगी केक करूँगा, yeast पकेगी mushroom करूँगा, yeast पकेगी तमें ये भी करूँगा, मतलब sauce भी करूँगा. sauce का होगा पता वास्ता होता है. ये टाइप of relationship मुझे तास नहीं लगता. Pseudomonas, Olispera and Rhodococcus. Olispera is bacteria acting on plastics. The name is Olispera castellatix. See, plastic factor is like one day one page in use. Yes. Now second one is Pseudomonas culture. Like Pseudomonas जहाँ भी culture. The DNA तो आने लगा. ठीक है सर, करो 90 to 90 degree Celsius. Yes. It is all 90 degree Celsius. ताकि तो anything करो anything Anything is that DNA primers are get attached to the single strand DNA. Correct. DNA primers are attaching at the 3' region. The पूरी गरम करो 70 degree Celsius. Yes. तब छाड़िए. Because high temperature लेने up to 90 to 94 degrees लेने से separation होता है. No. Tag polymerase, a specific polymerase being used here, whose name is thermos aquaticus, which has withstand temperature of 120 degrees Celsius. Then 90 to 94 degrees Celsius PCR form करें, सिर्फ DNA start correlate tag polymer construct करें enzyme protein tag nature ताकि ठंडा वाला बड़े tag polymer ना करें annealing करेंगे तीन स्ट्रैंड जो 50 to 60 डिवीजन है इस 72 जाए जाऊँगी फिर अटैच होगा प्राइमर एक्सटेंशन ऑफ हीट में ना ये आउ हीट होगा सिर्फ प्रोटीन नष्ट है अगर हीट शॉक दे ठीक है ये टॉपिक इज गुड
Next up, we have Shruti Patra of Stewart Science School. I request her to kindly come to the podium and present her presentation. Please welcome her with a round of applause.
Good morning, respected judges, teachers, and all my dear friends. It's a great honor to stand before you today in this competition. My name is Stewart from Standard 11 of Stewart Science College, and I'm here to represent my thoughts on the topic Chem Informatics, which is a field in bioinformatics. So first let us know, what is bioinformatics? If we divide the word bioinformatics into three halves, bio, inform and matics. Bio is for biology, inform is for information technology, and matics is for mathematics. Hence, bioinformatics is a branch of science which combines biology, computer and mathematics to analyze, predict the chemical sequences, genes and proteins. So now let's come on, what is Chem Informatics? Chem Informatics is a branch from bioinformatics which helps to predict genetics, which helps to predict our chemical data basically on drug discovery and chemical activities. It represents our molecular structures, managing chemical compounds. Its applications are wide in the field of medical science, like whether a compound is suitable to be a drug on the basis of toxicity, absorption, excretion, etc. It also discovers how two small molecules combines to form a larger and a complex stable molecules. We can see by using computational methods, it can predict the likelihood of a compound binding to target proteins, resulting in desired biological activities. By using computer-based techniques, we can predict how they can stable. Now, what are the advantages in this particular field? It enables rapid processing and analysis of large chemical data sets, facilitating insight and chemical structures and properties and reactions. It speeds up the identification rate of a compound, which is helpful for many researchers to search through massive database. Then, what are the tools that are used in this particular field? Tools like DevChem, ChemProp and AlphaFold use machine learning to predict the molecular binding affinities. Platforms like OpenAI and Article perform virtual screening of large chemical libraries to identify potential lead of a compound. Tools like RedMed generate new molecular structures for desired proteins. Now, what is the use in its current world? In COVID-19, we can see it predicted the inhibitors of SARS protein and their binding affinities. It also helped in cancer research by identifying anti-cancer compounds and predict their efficiency and toxicity. It had also helped in rapid identification of HIV and influenza, leading to successful development of drug. Last but not the least, I would end up by saying Chem Informatics is a very interesting field in bioinformatics. Chem Informatics bridges chemistry and informatics for more efficient drug discovery. Thus, it plays a vital role in shaping the future of medicines, biotechnology and computer, which unlocks the endless possibilities for human advancement. Thank you.
Any questions? Okay. This is a very interesting topic. Please remember, this is not a part of bioinformatics. It is a part where chemistry is added up with IT sector. Biology is bioinformatics, chemistry is cheminformatics. Even now, physics also coming. Due to that, we have empty simulation and all. Physics also coming. Okay. Now, my question is, why you not include smiles in this? You know smiles? SMILS?
No, sir.
It is simplified molecular input library system. When you are writing anything in computer, computer will never understand what is CH3, what is CH4, what is carbon dioxide. It will always understand a bit language. 1010 format.
Yes.
So when you do cheminformatics, we use a particular technique to give a chemical. Like if you have CH3, we write C111. So that the computer system will understand. That is how smiles. Anyhow, the topic is good. No more questions, right? So now we are ending with the higher secondary school plus 2 students, I guess.
With this, we conclude the poster presentations by plus 2 participants and now proceed to presentations by plus 3 and postgraduate participants. from Regional Institute of... We'll continue after 5 minutes. With this, Now may I request Ms. Anvesha Kiran Pradhan from Regional Institute of Education, NCRT Bhubaneswar, to come to the podium and present her presentation. Let's welcome her with a huge round of applause.
Very good afternoon to all. I am Anvesha Kiran Pradhan. I am from Regional Institute of Education, Bhubaneswar. I have prepared a poster on the topic GMO foods, in which we create genetically modified crops that have a long shelf life. So, as we know, after the harvest, the vegetables, they lose their shelf life very fast. So it is a very great problem. So we have to tackle this, and we can tackle this by this method, GMO foods, in which we use computation analysis, genetic databases of the crop, and we modify it to extend its shelf life. Here, bioinformatics and computational analysis play a crucial role by analyzing genomic, proteomic, or metabolomic data to identify genes and molecular pathways which control the post-harvest quality. So, here are some objectives that we can keep to identify genes related to freshness of that particular crop, to apply computational analysis or tools to integrate them, and to develop a newly formed genetically modified crop. So, I have taken the example of tomato. We can study we can see first we can identify the freshness related genes. There are so many genetic sequences that control the freshness in tomato. Then we integrate proteomic and metabolic data using computational analysis. Then we compare the genomics that control the shelf life of the crop, that may be long lasting or yes. So by using tools or databases like string, catch, metabol analysis, we can store or we can predict the sequence, and we can change or modify the genetic sequence of the crop. So, for conclusion, I am concluding my speech. Computational bioinformatics transforms vast biological data into actionable insights that enhance freshness, reduce waste, and ensure vegetables reach consumers in optimal conditions. So here I have used the term genome to market. It refers to the genetic informations or genetic researches we done in lab, and then we take it into the foods or crops to that gene, and we sell it to the market, which have a long shelf life, and it improves the nutritional values and great shelf life of that crop. Thank you.
Tell me what exactly you have done to find genetically modified food as a topic. Why it is required? Are we using it?
Yes.
How?
Sir, it is basically for the for extending the shelf life of the crop that are usually getting rotten in very short period. So for storage purpose or extend the shelf life, we can use this method.
Viksit Bharat, India is going to incorporate other from Russia. Okay, fine. Thank you.
Thank you, sir.
Thank you so much for your wonderful presentation. Now may I request Ms. Archana Sahu to come to the podium and present her presentation. Let's welcome her with a huge round of applause.
Good morning, respected judges, teachers, my seniors, juniors and my dear friends. I am Archana Sahu of Plus 3 first year from Computer Science Department of Stewart Science College, and today I am excited to present my e-poster named AI, the New Biologist. So, as we know, biology and technology are growing at a lightning speed. Every second, enormous amounts of biological data are being produced. Managing and analyzing the data, and that too manually, is a huge responsibility. And this is the point where artificial intelligence enters the scene. Now, as we know that before going into AI, let us first discuss what bioinformatics really is. Bioinformatics is a bridge between biology, computer science and mathematics that is used to analyze and interpret data. Now, coming to the role of artificial intelligence. As we know, genomic and disease prediction, AI can scan through millions of DNA within seconds and help us spot the disease mutating genes. Machine learning models help predict diseases like cancers, diabetes and genetic disorders long before the symptoms appear. In drug discovery and design, before, drug discovery used to take years, but now it takes few months. AI now simulates how molecules interact with human body, helping scientists find effective drugs in months rather than in years. Companies like Pfizer and Moderna are using AI in vaccine development. Protein structure prediction. Everyone has been knowing about the famous model, DeepMind's AlphaFold, and AI that predicts the 3D structure of the protein, that too with an amazing accuracy. This breakthrough helps researchers understand diseases at a molecular level and design better treatment. In precision and personalized medicine, AI also helps doctors design personalized plan based on each patient's DNA. It's like making medicines just for you, not for any average person. In big data and cloud integration, finally AI helps manage and analyze the massive biological data sets stored in clouds, allowing the scientists around the world to share their insights instantly. So now coming to how AI helped humans in different fields. The most recent example is COVID-19. As we know that the COVID-19 had single-stranded RNA as its genetic material. So the mutation was very fast. Now the AI entered the scene and helped the scientists to predict the mutation and even design the vaccines faster. Now coming to agriculture. In agriculture there are many variants of crops. Diseases are also huge and climates, it's changing. So in that field, artificial intelligence help the scientists or the researchers to make climate resilient and disease resistant crops. In healthcare sector, it helped doctors make personalized treatment plans. In environmental biology, as we know, 1.2 to 1.5 million species of organisms are present, but many of it are endangered. So to detect those manually is really a cumbersome task. But here artificial intelligence makes that easy by helping us through DNA samples to collect about the endangered species. At last but not the least, I would like to say that we humans are considered about only three things: why, what and how. Now, why AI? Not because I am a computer science student, but we are moving into an era where artificial intelligence will not overcome us, but empower us. Thank you.
Any questions?
about managing, analyzing data set. Can you tell me which one is the source for data set, biological data sets? From where you can get that data set?
Ma'am, when AI is taking all the data from our
No, just tell me the source of data set. Like suppose you are getting this from Google. So is there any source for getting biological data set? You all are telling about that prediction, analysis, data management, etc. So can you tell me any source of getting this data set?
Oh ma'am, sorry, I don't have that idea.
There are data sets, we have databases for all this analysis. I just want to ask one question. Differentiate between AI and algorithm based program. You are a computer student, no?
Yes, sir.
The differentiate between AI and algorithm based program. Why AI now is a boon during COVID, I guess.
Yes, sir.
But the AlphaFold, the deep mind learning library of AlphaFold, before that we had PDB, before that we had Uniprot. They were much before your AI.
Yes, sir.
So the accuracy and prediction were also there.
Yes, sir.
And also now. So there's only one question on mind, that what is the difference between AI and your algorithm based program or server or tool or software, what to say? And second is, if this is more accurate, then how we are expecting data access?
So I don't have any proper answer for this.
Actually, I don't know that. I heard you, but I thought after some time, because when you write that deep minds AlphaFold, no?
Yes, sir.
That is an AI based method, but it is not completely AI. It is
Yes, sir.
machine learning.
Yes, sir.
You have to prepare it. That's what kind of set, from where you get data set. You have to prepare data set.
Yes,
What
sir.
is your name? Tell me please.
Sir, Archana Sahu.
Archana Sahu. You please write your name in the AlphaFold search box. It will not show it.
Yes, sir.
So it recognizes some this certain patterns over there. It's 20 to the power 20.
Yes.
20 amino acid to the power 20 amino acids.
Yes, sir.
The value comes from 4 million or something like that. तार value से notes. तो तार छोड़ा कुछ powerful है. Okay, thank you.
Thank you, sir.
Thank you so much for your insightful presentation. Now I may request Ms. Asheen Fatima from Christ College to come to the podium and present her presentation. Let's welcome her with a huge round of applause.
Today's most emerging field is the field of research and molecular biology, that is use of artificial intelligence and machine learning in the prediction of protein structure. First of all, proteins. They are the most fundamental and the complex structures in our body that are responsible for various biochemical and metabolic reactions. But how they are functioning, that is based on the three-dimensional folds that they are producing us. And we can actually know the prediction of the protein structure by traditional methods such as X-ray crystallography, NMR and cryo-electron microscopy, but they are actually very time consuming and costly and use various laboratory techniques which we actually cannot afford. So at this point, the AI and machine learning comes to rescue. If we provide a perfect feed to the AI, they can generate a remarkable accurate result. So here the AlphaFold, Rose TTA fold and OpenFold are various AI and ML which helps in the prediction of protein structure. Basically, AlphaFold does the calculations of the distances and the angles between the protein folds, and it says the structure, the angles that are present, and if we identify that particular sequence, we can get the entire result of the protein. Like it basically uses a simple amino sequence to give the exact result of the protein structure. Now Rose TTA fold, it is a 3-track network in which it identifies all the primary, secondary and tertiary structures of the protein and gets fed into it. Generative models are now used to design various today's research. The key techniques that are used by this AI and ML are CNNs and transformers. CNNs are the convolutional neural networks. They basically scan and capture the structures of the protein, and we can refer to later. Transformers, long-range depictions between the first to the last of the protein structure. The whole of the protein structure we cannot study, but if you feed it in that, it will give a result later on. Attention mechanisms basically focuses on the higher weights of the protein sequences so that we can instantly get the result. And MSA integration, that is multiple sequence alignments of the proteins that are used for the evolutionary relationships. Like previously what the proteins were doing and now what the proteins are doing. And it is actually a very revolutionizing molecular biology topic in drug discovery and vaccine design, in which the targets, how the proteins are getting attached to the targets, this is used helpful in that AI. Enzyme engineering, how various enzymes are functioning to a particular protein, and we can also make enzyme based on the protein structure prediction. Understanding genetic diseases, that is how the mutational protein fold is different from the normal protein fold. If we see that, we can actually identify the drugs and the medicines to that diseases. So last, at least it's a long experimental thesis is converted into short, and our study approach is also very short if we use AI. So thank you.
Going ahead, I will first congratulate you that you know about MSA. Right?
Sir, actually, I am a zoology student, so I have done a lot of research on this particular topic because I didn't have any idea, so it sounded very beautiful to me about knowing that.
I can understand when you have built this structure. Otherwise, you will not be able to understand how you begin from first to last.
Sir, I know it is a simple topic, but I thought it will be easier to explain and.
That's simple, Nirina. It depends on you how much research you can do.
Yes, sir.
Understood. AI, the act of AI is everywhere, but how much, what is your perspective to look into it? That is what research is.
Yes.
Research is not something new. It is already been searched. Again, you are searching. That is
Yes.
just research.
Yes, sir.
Okay. My question is over here. If you take MSA, could you please tell me why it is yellow, purple, green, yellow over here?
Sir, if we see a protein structure in 2D, how we are studying in our textbooks, we cannot define the relationship between front to end. But if we see it in a 3D way, then we can actually predict that first ending to the last ending, it is actually connected like that.
Okay. Why it is taken vertically, not horizontally? You understand? See the numbers of A, C, P, R, L, D.
Yes, sir.
It is all vertical.
Sir, research has been done like that. If we take it horizontally, then we cannot. Maybe we can. Sir, I don't have idea about that.
See, when I take MSA, MSA means multiple sequence array.
Yes.
You have to take more than 2 sequences.
Yeah.
So that sequences what you have taken are all sequences. And the yellow, purple, green is showing that at this position they are same.
Yes, sir.
At this
Yes,
position
sir.
they are same. Depending on that position, the pink color fold you see, that is for alpha helical structure.
Yes, sir. Yes,
This is
sir.
occurring by.
Sir, can you please answer me that we can take it in horizontal form or not?
That is pairwise alignment.
Okay.
This is pairwise alignment, you go by strings.
Okay, sir.
So it comes in horizontal. But when you go by MS, multiple strings alignment, it comes in vertical. This is not blocks.
Thank you, sir.
Okay, what is CNN?
Sorry. Convolutional neural networks. They are basically the structures that
What
scan.
of the form?
Convolutional neural networks.
Yes. Can you define the term neural network?
Ma'am, that is basically the parts that are used in that particular technology that is used to scan the protein.
Just tell me the key definition of neural network.
Neural network, if I define it according to biology, then it's.
Okay, thank you.
Thank you, sir.
Thank you so much for your wonderful presentation. Now I would like to request Aurobindo Vinayagiri Goswami from Raghunath Jio Degree College to come to the podium and present his e-poster. Let's welcome him with a huge round of applause.
Good afternoon, everyone. My topic was biopolitics in software and tools. In this way, the agendas are definitions of biopolitics, history, objectives of biopolitics, tools of biopolitics, and applications.
Pila, e-poster competition. You have to present a poster.
It doesn't create a poster.
It's a presentation. It's a poster.
I know, but I doesn't have any time, so that I will.
अच्छा यार summary
So now summary is done. The 18 is done. These are in bioinformatics. We will use the biology and mathematics and data processing in the one. That there are the objects and technology we will use in bioinformatics to explain how biology is related to computer science and technology of the networks. There are the tools which are used in bioinformatics like databases, homology and similar tools, product functions, structural analysis, and sequence analysis tools. The biological databases. These databases usually consider genomics, proteins, and metabolic data. That data includes structure of ligands of amino acid structure. Some major biological databases are major nucleotides, sequence databases, major multiplication databases, major genes explanation databases, major microbial genomic database, major organism specific database, major proteins database. Homogeneous and similar tools, homogeneous sequences and sequences that are related by deficiencies from common acids. Thus, the degree of similarity between two sequences can be measured. This set of tools can be used to identify similarities between novel sequences of unknown structures and functions and data types, structures to structure and
from Stewart Science College to come to the podium and present his presentation. Let's welcome him with a huge round of applause.
Hello. Good afternoon to esteemed judges, respected teachers, and my dear colleagues. My name is Bharat Bhushan Raut from Stewart Science College, and I am currently doing my bachelor's in physics. Today I am going to tell on the topic bio-inspired humidity sensor based on diatom use R and electrolyte displacement. in which I am going to propose one of my conceptually invented device, a humidity sensor, whose functioning is to find the relative humidity of the atmosphere. So before going into it, let me give a awareness about bioinformatics. So bioinformatics traditionally is the data analysis of genes, protein and medical records, and like Sabir Sachi sir told that he is expert in drug exploration. So yes sir, you have said that you explore drugs. So these are all the famous things in bioinformatics. But the scope of bioinformatics is wide. It also contains the bio-inspired device or technologies that produce an enormous amount of data that can be analyzed using computational intelligence. So my project is based on such a fusion. So let me tell about the working of my device, which is a humidity sensor. So this is made up of two layers. The top layer is made up of a thin layer of Kisovo filled with electrolyte, and beneath it we place a solvent. Generally it is water, and we add some electrolyte into it so that we can eliminate some instrumental measurement error, that tiny measurement error. To eliminate that, we add electrolyte into it. So when we expose the first layer of the device to the atmosphere, then due to the water molecule present in the atmosphere, it displaces the electrolyte inside the Kisover. Since Kisover is made up of SiOH and water for hydrogen bonding with it, so it has an affinity to water. So we take a non-aqueous electrolyte for that reason, since non-aqueous electrolyte doesn't have hydrogen bonding with it. So whenever we expose the first layer of the device to the atmosphere, the water molecule displaces the electrolyte, and this electrolyte due to gravity fall into the solvent placed beneath it, increasing its conductivity. So we can measure this conductivity by passing electrical current through it. For example, if we get 1 microampere reading in the ammeter, then from that we can, from the graph in the top left corner, we get the relationship between current and conductivity of the solvent. So from that we get an equation which is y is approximately equal to x. So if the current is 1 micrometer, then the corresponding conductivity is around 0.2 mole per meter. So we can get by multiplying volume into it the mole of the electrolyte, which is if the mole of the electrolyte is 0.2, then if the container is 1 liter in volume, by multiplying that into this concentration term, we will get the total amount to be 0.2 mole. So the total amount that is displaced, the electrolyte which is displaced from the Kisover, that is the same amount that the water get absorbed into the Kisover. So if 0.2 mole of water electrolyte is displaced, then 0.2 mole of water get absorbed. So from the graph in the bottom left corner, we see a relation of y equal to some constant times log x plus constant. So if the absorption is 0.2 mole of water taking place, so in percentage wise it will be 20% somewhat. So generally time is up, so I will wind up it quickly. So I have actually devised an instrument which is a humidity sensor that is made up of, so in the innovation part is it is made up of two layers. The upper layer is a thin layer of reservoir, and below it we place a solvent. So whenever we expose that to the atmosphere, so the water molecule displaces the electrolyte, and the electrolyte increases the conductivity, and that increased conductivity can be measured to find the relative humidity of the atmosphere. So we can integrate this to the computational intelligence to study various biological systems such as dermatoinformatics. In dermatoinformatics, we can study the relation between the rash in skin and we can study the relation between rash in skin and the effective humidity of the atmosphere. Thank you, everyone.
Okay, any questions? What is Kissel?
Kissel is actually a fossilized porous substance.
It is a diatom.
Yes, it is a diatom.
It is
So,
a diatom, which means that
yes, sir.
they have two identities fused together to make a powdery substance.
Yes, sir.
Now you are talking about the cells that you want to treat, whether they are dividing or not. You are going to treat from that. The process what you have taken, we can use it for rainwater, but for the skin, or if you want to check out for a skin or dermatology or suppose cancer, you have to make a precise protocol for that. Why I said no? The diatoms are normally allergic to skin. ଆମର ତ ଜାଣିଥିବ ଆଗରୁ ଲୋକମାନେ ମୁଲତାନି ମିଟି ୟୁଜ କରୁଥିଲେ ଏସବୁ ବି ଡସ୍ ଥିଲା
Sir, actually, for the moment, for a cheap testing, I will use diatoms in my instrument. But whenever that experiment produce, when the data is sufficient, and I can represent to in a research paper. After that, I can make a synthetic microstructure replacing these diatoms.
You want to make an assay. It can be sold out. You can use it as a plastic cover. You can put a drop of water to indicate something. I understand it. But when you are talking for a biological treatment, you are doing like this, then you have to also understand the composition. We are saying about electrolyte, but what are you going to use for electrolyte? Now I'm saying, if I add sodium chloride water in Kissel wood, it will act as best food for fungi. So it's a you have to precisely clear for that.
Sir, basically we cannot add NaCl. You said that we can also add NaCl. So NaCl, for that to be electrolyte, we need that aqueous form, and it have affinity toward the Kissel because it will also form hydrogen bonding. So we can take NaCl. We need to take non-aqueous organic electrolyte. Yes, sir. We can take NaCl actually. For that, we have to take the aqueous form, and it will also form hydrogen bonding, and it will have the same affinity like that of water. So water molecule will not displace that electrolyte. So it will not work generally.
Okay, thank you.
Thank you, sir.
Thank you so much for your wonderful presentation. Now I may request Vidisha Mahanti from Ravenshaw University Mahanadi campus to come to the podium and present her presentation. Let's welcome her with a huge round of applause.
Good afternoon, everyone. I am Vidisha Mahanti, a second-year BCA student from Ravenshaw University, and my e-poster is on the topic Machine Learning for Disease Prediction. In our ancient scripture, Manusmriti, it is written that Arogyam Paramam Bhagyam Swastham Sarvartha Sadhanam. This verse means health is the greatest blessing. It is the foundation of achieving all the other goals in our life. From the wisdoms of Manusmriti to the algorithms of modern bioinformatics, the goal has remained always the same: to preserve life through knowledge. And in today's world, this goal continues with the help of technology, and one of them is machine learning. So now, what is the role of machine learning in disease prediction? It basically acts like an intelligent assistant to the doctors. It learns from thousands of patients' records and then it learns patterns and then it tries to predict the disease like diabetes and cancer and many more before they become life-threatening. Now moving on to the how this process actually works, it involves few steps and the first step is data collection. So basically in this step, the medical and biological data like DNA sequences and lab results are being collected from various sources. And then the second step is data pre-processing. Raw data often contains some errors and missing values. So in this step, the data is revised, filtered and then converted into a structured form. Then the third step is feature selection. In this step, the important attributes from the data is being collected. Then the fourth step is training and testing. This is the most important step in the entire process. In this step, a huge amount of structured data is being provided to the model and then the model analyzes it and learns the pattern and then after the completion of training process, the machine again given with new patient's data and then the machine tries to predict whether the patient has some disease or not and then its results are compared with the real medical results and then accuracy is measured. If the accuracy is low, then the model is retrained. Then the last step is evaluation and improvement. This step is almost similar to the testing step, but the only difference here is the results are analyzed and the algorithm is adjusted if it needs to be. Machine learning in healthcare has various benefits as some of them I listed here. As you can see, it detects early, it has higher accuracy rate and the most important thing is it saves doctors time. And it is already been applied in some areas like cancer detection, heart disease prediction, diabetes forecasting and many more. And that chart shows the accuracy of disease prediction using machine learning versus traditional methods. In the first part, you can see in predicting cancer disease using machine learning, the accuracy rate is 96%, and by traditional methods, it is only 70%. And in second two parts, we can see in predicting heart disease using machine learning, the accuracy rate is 88.5%, but by doing so using traditional diagnosis method, it is only 75%. Then in future scope, future with machine learning is quite exciting as we can expect it in wearable devices like smartwatches, where it will monitor our health record regularly, and we can also expect it to design our personalized treatment plans. And then the conclusion part, machine learning is not only transforming our healthcare system, it is also saving our lives. So as we are moving forward, this bond between healthcare and human well-being and technology will lead us to a healthier and smart future for sure. Thank you.
Can you define data pre-processing? What do you mean by data pre-processing?
Ma'am, actually, when we collect data, they are very raw.
And just give me one example of data pre-processing.
Okay, ma'am.
You are writing that data cleaning. Can you give some example?
Ma'am, raw data may contain some errors. Like if we are collecting some data, there may be some printing mistake. If the patient's age, let's guess, 50.
When we are collecting data, the data is stored in which format? Excel store korcho ki Q3 store korcho. Just one example there.
Database. Using database. Actually,
okay, printing mistake. Printing mistake is data. Error means printing mistake.
No, ma'am. That's not my point. Ma'am, let it be 50, but by printing mistake it is 500. So during this step, this
error is rectified, and during this step, the testing data, the data is divided into training set and testing set. Both are different, and also in during this process.
No, that is training. Training set and testing set is in training and testing.
Yes,
80-20 rule. No, I am just telling that you are writing pre-processing. What is that error? Printing mistake is the error.
No, ma'am. During this step, the data is converted into the machine understanding format.
Okay, so you are saying that accuracy comparison when you use the AI method is more than the traditional method. Your prediction says over there that accuracy comparison when compared, AI method is more likely to be correct than your traditional method, correct?
Yes.
Just tell me one thing, the same error what you are saying, if the error goes on your AI machine and predicts something very vast, which is not at all accepted by your human body, what check you have to do?
So I didn't get your question.
I'll let you know. You have a heartbeat. What is the rate of heartbeat?
Sorry, sir.
72.
I'm unaware.
72 is a heartbeat. And you have a machine called as ECG, electrocardiogram, which is not AI based. Now I replace ECG with the AI based machine, and it reads that your heartbeat is somewhere 700 per minute. Typing error. डॉक्टर तो उस मैनुअल डॉक्टर आईसीयू में एडमिट कर दो कोई बोल
Sir, this rectification, this such kind of rectification is done during the data pre-processing. Like I was trying to say to ma'am, a human can live to 500 years. It is the
that is correct. What you are saying, understand. Data pre-processing is like a screening. You screen out which one is essential for you, which is not essential to you. You keep it for set 2 or something else. Even more by that, when you are using doctor to predict disease on the basis of accuracy comparison, do AI have any limitation? If yes, then what is the backup plan? In your Play Store, there is an app called as Arogyam, AAROYAGM. Everyone can use that. You put your fingerprint, you put your you put the app on a specific part, it will show you the result. But don't go by that.
Yes, sir. Machine learning has some limitations, and it is only solved during training by providing it quality training data.
Okay.
Thank you, sir.
Thank you so much for your wonderful presentation. Now I will request Devanshi Kanda from Saravana Women's Autonomous College to come to the podium and present her presentation. Let's welcome her with a huge round of applause.
Good afternoon, everyone. At first, bioinformatics means the branch of science where biology meets information technology that helps us to understand the biological complexities. In bioinformatics, it uses the computational intelligence like artificial intelligence and many more algorithms and databases to understand biology. Here we can see that there is a DNA sequence, chemical cells, molecules, bacteria, fungi, etc. and also chemical molecules and various things are here. And the human brain and the computer brain, it is very similar, like how human brain helps us to understand things we do on our daily life, as well as computer brain helps us to understand the things computer does. For humans, just like human brain is main, computer brain is also made for the computer. it helps us in analyzing data and how bioinformatics helps us to store helps us in various things. First of all, it helps us data collection. We can collect data of various people, and then there is data storage, and we have to store the data in large databases, etc. Then is data interpretation. We have to interpret and analyze the data we have stored, like how a protein works or how the gene expression are coded. Then there are various applications of bioinformatics, like decoding genes and understanding how a gene works. We can also do it that understanding genes and diseases and agricultural improvements, etc. Thank you.
Any questions? Did I have drawn this one?
Yes, sir.
Beautiful painting.
Thank you, sir.
But we have to give a poster, no? Where you should have an idea or an objective, a conclusion.
Yes, sir. I thought it will be quite impressive if I drew it.
It is quite impressive, right? Yes. Okay, thank you.
Thank you, sir.
Thank you so much for your presentation. Now I may request Diksha Sen from Stewart Science College to come to the podium and present a presentation. Let's welcome her with a huge round of applause.
Good morning, everyone. Today I am going to introduce you into the fascinating world of sugarcane omics and how bioinformatics transforms sugarcane from being a simple source of sugar into a smart bioresource for the future. We all know sugarcane as the plant that sweetens our tea and produces bioenergy. But what we don't know is that sugarcane has the most complex genome among plants, about 10 billion base pair long and highly polyploid. In order to study such a complex plant, scientists rely on the integration of omics technology and bioinformatic tools. Now, what is omics? Omics is the study of life at different stages in the molecular level. It comprises of genomics, transcriptomics, proteomics, epigenomics and metabolomics. But these allow us to give us a lot of information. So the data generated from these studies are analyzed and organized, analyzed and interpreted by bioinformatic tools. Bioinformatics basically acts as the brain of modern biology. Now let us understand how bioinformatics integrates with each of these omic levels. Genomics. Genomics is like basically cracking the code to the sweetness of sugarcane. Imagine the sugarcane's genome like a gigantic library, about 10 billion letters long and filled with multiple overlapping copies of the same kinds of books. So bioinformatic tools basically allows us to read, sort and annotate this massive collection. With NGS, next generation sequencing and bioinformatic tools like BLAST, Maker and BlowTie allows us to identify which genes are responsible for high sucrose content, disease-resistant varieties and high fiber quality. Comparative genomics allows us to compare the genome of sugarcane with related varieties like sorghum and maize. So we can identify which genes are shared and which genes are unique to sugarcane. All of this data is stored in online databases like Sugarcane Genome Hub and NCBI GenBank. So bioinformatics acts like a librarian that keeps this massive genetic library organized, searchable and useful. The next one is transcription.
which genes are switched on or switched off during different conditions like stress, drought, growth periods, or even during sugar production. Bioinformatic tools like Trinity, cufflinks, these and two help us to assemble the RNA data and to identify differentially expressed genes. For example, during drought, certain aquaporin genes become more active in order to conserve water. So bioinformatics and transcriptomics allows us to hear sugarcane's molecular voice of survival. The next one is proteomics. Proteomics is the machinery behind the sweetness. If genes just gives us the instruction manual, the proteins are the machines that make things happen. Using 2D gel electrophoresis, LCMS, scientists can identify which proteins are made and the amount of proteins made. Using bioinformatic tools like Mascot, UniProt and String, scientists can make protein-protein interaction networks. So bioinformatics and proteins is a very crucial integration method. Bioinformatics acts like the engineer that studies how proteins work together to make sucrose. The next one is metabolomics. Metabolomics is the chemistry behind the sweetness. Metabolomics focuses on the final chemical products of plant, that is the sugars, amino acids, etc. and other metabolites. So this basically is responsible for the flavor and energy and yield of sugarcane plant. So the bioinformatic tools like MetaboAnalyst, Cytoscape and KHEG pathway helps us to link certain traits, certain metabolites to particular genes or proteins. So metabolomics and bioinformatics together tells us why certain sugarcane plants are sweeter than the others. The last one is epigenomics. Epigenomics is the memory of the plant. This is where things get really fascinating. Sugarcane actually has some kind of a molecular memory. Using DNA methylation and histone modification, sugarcane can remember past stresses like drought, heat. Bioinformatic tools like Bismarck and Metco can identify these epigenetic patterns in the sugarcane genome. So this actually enables us to learn how the sugarcane adapts to different environmental conditions just like we humans do, and the memory we retain. Lastly, the integration of bioinformatics. As the time is up, I will just conclude it by simply stating the applications. So this technology enables scientists to build high disease-resistant varieties of sugarcane, and CRISPR-Cas9 gene editing technology, which is the latest gene editing technology, allows us for precise breeding. So this is to sum it up, sugarcaneomics provides us the data, bioinformatic tools gives it its meaning, and together they are shaping a smart future for plant biology and biotechnology. Thank you.
Who gave you this idea?
So this topic was suggested by my teacher,
Who?
Ragishi ma'am.
Okay. You should give a round of applause for her. This is a very current topic on which India is going to work on. Not only for that, because molasses are also going to be used as fuel very soon. Molasses में जो पत्थर बारूदी
After juice extraction.
it is going to be used for two things. One is for cash crop of mushroom group and second is for fuel. Very good. अब देखो ओमिक्स से बाहर कर रहे थे वो कौन विस्तार कर रहे हैं? प्रोटेमिक्स बाहर कर रहे हैं?
Sir, genomics, transcriptomics, proteomics, epigenomics and metabolomics.
Relationship कौन है?
Sir, basically these are separate omic fields, but my idea is to integrate all of these fields. Like
if you want to integrate, you should have one common topic. ସାପ ବି reptile, ଏଣ୍ଡୁଅ ବି reptile?
Yes, sir.
ମୁଁ ଆପଣ ସାପ ଏଣ୍ଡୁଅ କୁ indicate କରିଦେବି।
Sir, I can't really understand.
What I am saying is, what particular part from the sugarcane you have found in common while you are omics study?
But the source, like the DNA extracted from the sugarcane plant, for each of these omics there are different, different databases, and I'm extracting the DNA from different places.
Okay, so you have taken the sequence.
Yes, sir.
From different databases?
Sir, if I want to study like the sucrose content of the plant, then I would ideally take the DNA from the stem, because sugarcane is a modified shoot. It is a pulp, so the sucrose content in the stem is high. So it is ideal for me to take the DNA from the stem.
Other part of this program from where you take DNA?
Sir, I can take it from root and leaves. For leaves, if I want to study about the photosynthesis related activities, I can take the DNA from the leaf.
DNA is a photosynthesis method.
Sir, it's like how.
Oh, thank you.
Thank you so much for your insightful presentation. Now I may request Kamalakanta Raut and Rishikant Panda from Ajay Vinay Institute of Technology to come to the podium and present their presentation. Let's welcome them with a huge round of applause. Dikshant, then you gave right now. So after them, Kamalakanta Raut and Rishikant Panda, they both will give the one presentation.
A very warm good afternoon to all the respected judges, faculty members, and everyone present here. I am Rishikant Panda, along with Kamalakanta Raut. We are from Department of Computer Science, AVIT, going to present our poster on the topic that is impact of bioinformatics on AI-based automation in robotics. First of all, I will tell you about what is bioinformatics. Bioinformatics is a field that mainly uses biological knowledge for analysis of the biological data, which is now inspiring the main motto of my making this poster is that it bridges the gap between artificial intelligence as well as biological intelligence. By using the biological information like the genomic data and swarm intelligence and neural network, these can integrate with our robots and make it more smarter, reliable and adaptability. And it can evolve our living organization. The main significance of our making this project is that the traditional AI models are different from our biological based robots. The bioinformatics data set gives us deep insight about the biological systems like the genomic evaluation, neural learning, and swarm behavior. By applying these biological concepts and principles, we can create more efficient, adaptive, and intelligent robots. These have a strong implication on our healthcare, industries, automations, and environmental monitoring. The main methodology part that is I am going to discuss is that first of all we collect all the biological data like genomic data, protein synthesis, and neural network. These data sets are then processed. Then we applied some machine learning algorithms. After applying machine learning algorithms, we trained our AI models so that they can become more adaptive to environmental behaviors. Then for evaluation and testing performance, we will use our bioinformatic based robot to show more efficiency and adaptability. Now my friend Kamalaganda will explain about what the result and conclusion are.
Efficiency and capability in robotics, and genetic model optimize the pathfinding. As a conclusion, biotechnology inspires the algorithm and adaptability, property of the automation, bio-inspiration algorithms, the synergy of biology and AI, next gen intelligence, future robotics that can adapt and evolve like human beings. Thank you, sir.
Any questions? Okay, let me ask. Just tell me the impact of bioinformatics on AI-based automation in robotics. What is the motto?
Our main motto is that all are using AI robot for their daily uses, but they are not self-efficient. We use some biological data set like genomic data, protein synthesis data, and neural network data. These data are when fed into our robot, they become more smart.
Okay, that is what I am asking. What your robot is aiming to do? What he will do? He will operate, he will give medicine, he will kill someone, he will talk to someone.
For this, I would like to say that our robot will play a major role in healthcare and industrial part.
What?
Analysis करियो.
Analysis करियो. Analysis के robotics से पक्के analysis के भी नहीं करियो. वो क्या बोलते हैं? So what will be data equation collected biological data from genomics, proteomics, and neuromethods, right? Genomics, proteomics, and neuromethods. So you have a connection between a backend algorithm with the two separate identities. Genomics different, proteomics different. Then you write converted biological patterns into computational format for AI model. I understand. You take a sequence, you convert it into 1010 format, a binary format, one. Design bio-inspired algorithms like genetic, neural, and swarm models.
First,
can you develop genetic algorithm?
We will collect this dataset from certain websites like GitHubs and DAXoff. Then after that, collecting this dataset, we will use some machine learning concept and provide it to our robot. After giving those dataset, it will analyze using Python and some coding concept what is more accurate, and it will increase the precision of the robot in performing certain automations.
I understand, but you try to understand my point. If bioinformatics is analysis of system, right? Bioinformatics cannot cut anyone. Bioinformatics cannot operate someone. You only give an analysis and prediction. भाई तो एक आगू जाओ तो Google Map, right? 500 मीटर पर right to मिल जाती है. Bioinformatics involved in that format for biological systems. Now when I'm putting it into robotics, what exactly the robot will It will analyze.
It will analyze and help all the doctors and to do that operation.
Thank you.
Thank you, sir.
Thank you so much for your presentation. Now I may request Kritika Mahanta from Reverend University to come to the podium and present her presentation. Let's welcome her with a huge round of applause.
In India, there are 2.5 million people living with disease cancer. Every year, new cancer patients registered over 7 lakh. Cancer-related deaths are around 5,56,400. One woman dies of cervical cancer in 8 minutes in India. As many as 2,500 persons dies every day due to tobacco-related diseases. A very good afternoon to all. I am Kritika, pursuing BCA in Ravenshaw University. I am very glad to represent my topic, bioinformatics in cancer research and early detection. There is no doubt that cancer is very deadly. In US, it is considered as the second most cause of death after heart disease. Even after treatment and diagnosis, cancer is potential to kill. So, before starting, what is bioinformatics? Day by day, our researchers and our doctors inventing new technology to save lives. From those things in this AI world, bioinformatics is one of them. So first, what is bioinformatics? Bioinformatics is the use of computers and software to analyze biological data like DNA and proteins. It helps scientists understand disease, develop new medicines, and make biological research faster and more accurate. Then, what are the objectives of bioinformatics? There are 3 objectives I mentioned. First, store and manage large biological data. Bioinformatics plays a great role in storing, maintaining, and analyzing the large and massive amount of biological information generated by modern research. Technology like DNA sequencing and protein sequence analysis, it is hard to maintain by human beings. Then there are many real-life applications such as drug discovery, vaccine discovery, early treatment, etc. Then I mentioned the major branches of bioinformatics such as data management, sequence analysis, structural bioinformatics, computational genomics, and system biology. Then we will dig into a topic that cancer research and early detection. How bioinformatics help in cancer research. So first, what happens in cancer? Cancer results from genetic mutation that cause uncontrolled cell growth. Detecting this mutation early is critical for saving lives. Bioinformatics plays a major role by analyzing large-scale biological data using advanced computational tools to understand cancer at the molecular level. In this table, I mentioned how bioinformatics helps, like in which area bioinformatics help, how it does, and what are the benefits we got. Like first, in cancer gene identification, normally, basically it happens that in which gene actually mutation happened, through it we can understand the cause of cancer. Second, tumor classification. Sometimes we think the symptoms is similar, like cancer, and we think that is cancer, but actually that is not cancer, so we can identify which is tumor cell and which is cancerous cell. Similarly, there are lots of area where we can take help from AI, like a biomarker discovery, targeted drug development, survival and risk prediction, and data integration. Then, as I mentioned, application like detect cancer-related mutation, identifies drug targets and biomarkers, design personal cancer drug. As every machine and every technology have some advantages and disadvantages. In advantage, we have early detection, personalized treatment, fast analysis, drug discovery. So disadvantage like high cost, some people can't even afford, and this is very the treatment is very high cost. Then data privacy, hard to interrupt, and limited access.
AI help in early detection, then what are the methods through which we can detect it early, like liquid biopsy, genetic screening, imaging, bioinform, risk analysis, biomarkers, and the last AI and ML. At the end, the conclusion is about bioinformatics accelerate early cancer detection by analyzing complex biological data, enabling timely diagnosis, personalized treatment, and improved patient survival. Thank you.
Okay, anybody? Just tell me one thing. You said bioinformatics is used in cancer research, early detection. How?
So normally patients were diagnosed after stage 3 or 4, which is hard for treatment. Like at that time, doctor.
Stage 3 4 बोलते हैं इतनी चले जाना है कौन एनालिसिस नहीं करेगी stage 3 4 बोलते हैं इतनी चलती हो तो एनालिसिस क्या करेगी
So normally sometimes both later detect करिवे
exactly ମୁଁ କହୁଛି ବାୟୋଇନଫର୍ମେଟିକ୍ସ ନା ତ ଡିଟେକ୍ସନ କହୁଛି କେମିତି
जो म्यूटेशंस या जो कि एआई स्कूल में कोई जीन एक्चुअली म्यूटेट होला ये टाइम हम जान रहा कि एक म्यूटेशन है तो ये कैंसर कोज कर पाएगा एंड तब हमें अर्ली डिटेक्ट कर पाएगा एंड दैट ट्रीटमेंट कर पाएगा
सामान्य इट इज गोइंग टू स्टडी अ पैटर्न
यस
clear if the pattern had to be there then the pattern will say what amount of cancer is there okay then your time सब कुछ बोलते हैं ना हम बेटा ये तो वेरे तो
Thank you so much for your presentation. With this, we conclude the first half of our competition. We now break for lunch. Everyone is requested to proceed to the new block 2 and enjoy the meal arranged for you. And I would like to request our volunteers to kindly escort our guests for lunch. We also request